There we go. So synthesis step is when the virus is going to use its genome to take over the host cell. This is going to be happening in the cytoplasm of the host cell, which we can see a little bit on this picture. Big picture. There you go. So we can see on this picture that here's the virus now inside the host cell because it attached and it penetrated. And now, whatever genome it's got, whether it's RNA, DNA, double-stranded, single-stranded, the genome is going to take over the host cell. And then the host cell is forced to make new virus components. So the virus is not doing synthesis, it's the host cell making viral parts. So, you know, there could be 5 kinds of components depending on the virus. Any of them that the virus needs, the host cell is going to make those components. And the host cell, for it to do that, what that means is the host cell uses its own energy, uses its own building blocks, and the host cell has not much left for itself. It's doing the work for the virus. So in this example, here's the genome, and the genome causes the host cell to build new spikes and new capsid proteins and new genomes. And all of that takes ATP, and it takes amino acids, and it takes RNA bases and all of that. So it's taking up all the stuff and all the time and all the energy of the host cell. And the host cell might die from this if it runs out of energy and building blocks, but it's definitely going to be strained by this. And then the 4th step is, okay, now that the host has made all the new virus parts for the virus, they are going to do assembly. That's step 4. Step 4 is assembly. All the parts will come together and form a new virus, new viruses. So in assembly step, the parts arrange themselves into new viruses. And that's kind of shown right here. So here's the parts. They self-assemble into a new virus particle. So we see the genome, and then you've got the capsid proteins. And this one is enveloped. It's going to pick up the envelope on step 5. So I'm going to erase some of this stuff before I write down step 5. Make sure everybody's got this. I'll just erase part of it. How about that? So, okay, there's a new virus, viruses inside the cytoplasm, and they have to leave the host cell before they can infect another one. So the last step at the very end is called release, the release step. And this is when we get complete, finished viruses. We're going to leave the host cell, and they would be ready to infect new cells now during the release step. They have to get out of the host cell. So when we have coronavirus and we spread it to somebody else, what's actually spreading is virus cells that have come out of our nose or mouth epithelium. They bursted out of those cells, and they're kind of floating around in liquids of our mucous membrane, and then when we breathe them out. They would have released out of the cells and be in mucus, and then we breathe them out. The release would have come first. Release happens in two different ways, two options, depending on the virus. If the virus is enveloped, it's going to bud off the host cell, and that's shown right here. This segment of the virus is finished. It's got genome, it's got protein capsid, and it's going to pick up an envelope as it leaves the host, like wrapping around itself. So it's doing exocytosis. It picks up the envelope, and also this one is picking up. Sorry, it always makes me sick when I'm a student and that happens. It's picking up the spikes that are already in the membrane that's going to become its envelope. So this virus, here's the spikes made during synthesis. The spikes get sent out to the membrane, and they're waiting out here. And then when the virus exits the cell, it has some membrane with the spikes wrapped around it. That's how an enveloped virus leaves, by budding off the cell. If the virus has no envelope, or if it's naked, it lyses the host cell, completely breaks open the host cell and leaves that way, because it doesn't need an envelope. It's got to get out somehow, so it's going to break open the host cell to do that. This one, I want the lights all the way off, since it's black and white. This is one of my favorite pictures in all of virology. So what you're looking at here, we got this outline that kind of looks like an oval. And if you've got this picture, indicate that this is an E. coli cell. So we've got a rod-shaped bacterium. This is the cell envelope of an E. coli cell. And all of these blobby things that are all over the surface of it are viruses that infect bacteria. They're called bacteriophages. So it's a virus that infects bacteria. These have the complex shape. So if we go back and look at the shapes, their real shape is like this. If you ever come to my office, you can see my bacteriophage doll. It's both dorky and cute. It's one of those giant plush microbes. So this E. coli, notice it's got this gap up here. All of these blobs inside are viruses that are bursting out of it. And they're just kind of going to drift away. They can't move themselves. And even as this cell is dying, here's all of these other ones that are lined up out here trying to infect it. It's kind of like terrifying and awesome at the the same time. This is lysis of a host cell. We'll have a day on Monday that's all about coronavirus all day. It doesn't lyse cells, it buds because it's got a membrane. So it's not quite as horrifying, but two ways that viruses can leave cells. So one of the things that you want to have memorized for our next lecture exam is what are the stages? What are the steps in order? So we've got attachment, penetration, synthesis, assembly, release. 5 things. There's also 5 viral parts. You need to know those also. Those are going to be like list the 5 whatevers and then answer some questions about those. This slide is showing you how an enveloped virus penetrates a cell. So it does that by membrane fusion. So membranes touch each other after you have the spikes attach. And this is endocytosis, how the virus gets into the cell. Mm-hmm.
So why would a cell allow a virus in?
Why would our cells allow the virus in? That's such a good question. Like, why don't we just stop this? Why don't we get rid, I'm gonna go back a few slides. Like if this is letting the virus in, why don't we just not make that? That's, and through evolutionary time, like that's a big, if this is letting us be attacked and destroyed by viruses, why don't the people who had mutations in that like be the ones who live better and we get rid of virus systems? The receptor is always necessary. It's something that we can't live without. So for example, the receptor for coronavirus entry is really important to helping us control our blood pressure and all this other vital stuff. So it doesn't, whatever the receptor is, it's not just like, hey, I'm a virus, come over here. I'm for letting viruses in. It has a different job that's essential for life. But that's such a good question.
So it's like like our cells don't really have a choice.
Well, I mean, you could get rid of this, but you would die from it. Yeah. And it lets a virus in, and most viruses are not 100% deadly.
Okay, so like our cells don't have a choice on whether or not one can attach and one can't?
In general. So there are also, though, there are probably, have you ever heard of people being HIV resistant? What that is their receptor is just enough different to keep doing its normal job, but not bind on to HIV virus.
So like when you're like 12, 13 years old, a lot of people get like the HIV like vaccine.
No, that's HPV.
HPV.
Yeah.
So like how does that change our ability to resist?
That's going to make more sense after I get a few slides ahead. Yeah. So right now we don't have a HIV AIDS vaccine, but there is the HPV one. And that one, once I get a few slides ahead, it'll make more sense what my answer to that. Yeah. These 3 pictures are just better close-up pictures, views of what's happening at different steps. I'm not going to say more things about them really. They're visible. Like this is showing a particular enveloped virus that has these green spikes, how it leaves the cell. And kind of showing you, this is how big the virus is, and this is how big the cell is. And you can see that right now at this moment, there are 3 budding off. And here's some that just came from there.
So, like when there's there's not spikes naturally on the cell. It's when they have to make the other pieces is when the spikes show up on the membrane.
Yeah, so the spikes are not something that the host cell normally would make unless the virus told it to. And see how the spikes are kind of on this side? The spikes, the host cell makes the spikes. Let me see if I've got one picture, they're so tiny, but the host cell makes the spikes, and then the host machinery sends them out here. Like Jackie, we'll talk about that in cell biology, like how that happens, but the spikes are viral spikes. The host makes them. The host sends them out there, and then they're kind of ready for when the virus is going to leave. Did that answer kind of what you mean? Yeah. There they go. So imagine one virus infected this cell. How many do we have coming out in this view? I'm bad at counting things quickly. Is it like 13 or something? 12 or 13? Just coming out this side. So you had one land, 13 come out. That becomes important later. So this next two slides are kind of weird. They go together like they're horizontal. So see how this says 4? And this says 3. This was a huge, long, horizontal figure from your book, and I had to break it up onto 2 separate slides. And this is referencing 2 different ways that viruses can complete their life cycle. There's 2 versions of virus life cycle called lytic and lysogenic. I want to talk about lytic first. Lytic is the top row, and it includes all the steps that we already talked about. This is showing E. coli being infected by one of those bacteriophages. So you have adsorption, the virus attaches, penetration, at least the genome goes in. Here's the viral DNA going in. Synthesis is this. So if you've got this slide, just write synthesis right here above number 3. And now we're going to the next slide over. Now that we've got the viral parts, they will self-assemble. This step we don't include in what I teach. Maturation, skip over that. And then release. So this type of virus has to burst out of the cell, and there they come, and it looks like space aliens or something. Scary. And the cell has basically died from this. So all of those steps that we talked about, that's lytic cycle. And the majority of viruses do this cycle, or this style of life cycle. So all the steps are going to be completed. All the different 5 steps are going to be completed, let's say, quickly. The virus is not waiting around for the next step. It lands, it penetrates, it forces synthesis, it self-assembles, and then it releases from the cell. So that's lytic cycle. All of that's happening in a row, let's say.
So how quickly does this happen? Like in real time?
It totally depends on the virus, how quickly it happens. So it can be 25 minutes. It could be 7 days. It totally depends on the virus. But it's pretty quick. So if you ever, like before COVID, you get up and you go to school and you feel good, like in the morning, like, yeah, I feel good. And then at noon, you kind of feel off. And then at 1:30, you're like, oh man, I'm really sick. That's usually a viral infection because they tend to go quick. And then, if you have one land on a cell and 27 come out and they go and infect 27 more cells, you get really sick really fast. Tends to be. But it depends on the virus. So, most viruses act that way. There are some viruses that can do a different kind of lifestyle called lysogenic cycle or lysogenic state. And what's key about this is that the virus can insert its genes into the host genome. And this is usually going to happen sometime before synthesis. So it attaches, it penetrates, but then before any viral parts get made, the viral DNA, which is dark purple here, combines together with the host DNA. And it becomes a part of your genome. And this is this is scary, but this is worse because you've got viral genes in you forever now. Whoa. Fortunately, not all viruses can do this. Only some. Only some viruses do this. Not all of them. Not coronavirus. Thank God. One good thing about it. Some of the viruses that do this are HIV, which causes AIDS. HPV, which causes genital warts and ovarian cancer. No, sorry, cervical cancer. Those aren't the same. Cervical cancer. Those are two examples of human viruses that act like this. So once the viral genes are in with your DNA, they can sit there for some amount of time. That's what lysogeny is. You've got viral genes in with your genome, and those genes are just sitting there in your genome. And sometime later, some time passes, and you don't have any symptoms of the virus because it's not synthesizing, it's not making new virus, it's not killing your cells. You just got viral genes sitting in your genome. And then it's going to reactivate, synthesize, etc. And then you get comes at a way later time. So if you've got these slides, draw an arrow here to here. At some point, the virus will stop being in lysogenic, kind of dormant state, and it will go back into synthesis, assembly, release, and start doing the rest of the life cycle. So viruses that cause this kind of lysogenic state, you're infected, but you don't have any symptoms for some amount of time. And then later you do, way after you caught the virus. That's the worst. That's the worst. And there is a vaccine for HPV now, not yet for HIV. And it's because they both do lysogeny, but they have different kinds of synthesis strategies. Something, circle here, cancer. Viruses that do this tend to cause cancer because those viral genes are going to land in some random spot in our genome. And they are going to reduce the function, most of the time, of our genes. And we have so many genes that control cell division that it's kind of likely that when a virus goes and inserts into your genome, it's going to land somewhere that has something to do with cancer, and then you get cancer. Yeah.
Oh, you're writing that.
Writing what?
Oh, what you just wrote. Is that gold?
Yeah, that's worth knowing. Yeah. This goes with why cancer is caused. These go together. So HPV vaccine that teens can get, it's not actually, I mean, it does reduce genital warts, but that's not why genital warts are not a huge problem. It's kind of gross, I guess, but the cervical cancer is the deadly part. And so girls especially, really good to get this vaccine because it will prevent you from getting cervical cancer, which kills a certain number of women every year. European countries started doing HPV vaccine about 10 years before the US, and they now can see the effects, like cervical cancer has gone down in women there. That's how that works. So here's a better picture of lysogeny or lysogenic state and how it could cause cancer. So once in a while, the virus gene has actually something in it that will make cancer more likely. The other situation could be that the virus gene lands somewhere in our genome and messes up a protein that normally prevents cancer in us. Either of those can cause higher risk of cancer in people that have lysogenic virus infections. One other word to write, I forgot. There's a class of viruses that do lysogenic state that are called retroviruses. As a group, they're called retroviruses. So HIV, HPV are examples of those. Retrovirus, because by the time you see the effects, it was something that you got the infection a long time ago. This slide we're going to skip over. I'm going to erase this. We don't study viruses in our lab because they're a little bit harder to grow. Viruses won't grow on agar. You can't grow them in a broth. They have to grow in a host cell. So that makes them a little bit harder to study. In the older days, one of the best ways to grow viruses was in an egg. So you see someone here taking a really small needle and they're squirting viruses into eggs. And the eggs have host cells of a chicken that can then grow a lot of virus. You could do the same with a mouse, a dog, a person, but that's more unethical. You could also grow viruses in what's called tissue culture system. And these days, a lot of virus research is done in a tissue culture system where you have petri dish with agar, and in the agar you have a layer of animal cells. Animal cells is layer over the surface of the agar. And it's not an animal, it's cells that are growing in an artificial system, animal cells. That's tissue culture. So you could grow viruses in tissue culture. But to get them to do the whole cycle, you've got to have a cell for them to assemble in. That's how they assemble and how they complete their life cycle. That makes them a little bit harder to study than bacteria, let's say. It used to be that old school vaccines sometimes were made out of virus parts that came from eggs, and that people who were allergic to eggs couldn't get certain vaccines. That's less common these days, but that's where that concept comes from of people with egg allergies can't get vaccines. What we're moving into next is information about influenza. Influenza, up until the past 2 years, is like the most common virus that makes people really sick in Western Hemisphere. So I am still going to teach you some stuff about influenza in a shortened time frame because it helps understand things about coronavirus that we'll talk about on Monday. And I usually use this as the starting out question. Just last week, the U.S. Surpassed the number of deaths in the 1918 flu pandemic. That used to be the champion of killer virus for the U.S. Last week, or maybe it was this week, on Monday or something, we surpassed that for coronavirus. It's like crazy. But there have been a lot of deadly influenza outbreaks through the years in all different places. And they're all a particular type of virus called influenza, but they have different names of their subtype. So these would be like subspecies or versions of influenza. They're named with these particular names. And this one you would pronounce it H2N2. So where do we get those names? Flu virus has envelope and spikes and genome, of course, and capsid proteins. And flu virus has two different types of spikes. Let's call it influenza. Influenza viruses have two major spike types that are called H and N. H and N. I'm going to come back to this slide and I'll show you the well, I don't have a good picture of the H and N yet, but they have two spike types. The spikes control what cells can be infected. Notice on here, there was one called swine flu, like from pigs. There was one a few years ago that was bird flu. Sometimes you hear about bird flu. What influenza infects depends on the spikes. So the spikes control the host range for what animals can be infected. Here's another cool fact about spikes. If an animal has been infected and it has survived, it will normally develop immunity to that particular spike. Or let's say to that exact version of the virus. Like for me right now, I know I had COVID at the start of the semester. And if COVID acts like a normal virus, I ought to be immune to it, to that particular exact version, which is probably Delta, that I just had, because I survived. But the problem is, if you have a mutation, mutations make the viruses different. They're not the exact same version. And some mutations make them different enough that we're not immune anymore. That's a huge, big deal. Such a big deal. And later when we talk about vaccines, I'll do like a hilarious example of how this works in your body. But if you can survive a virus or any other pathogen, and you live and your immune system works normally, you're going to be immune to it. Unless it mutates and it changes into something so different that your immune system's like, nope, that's something different. Never seen that before. Dang. So we'll get to that later. But that's a huge deal for flu vaccine or influenza. Before I go to this, these subtypes called H and N, they're named after the version of H spike and the version of N spike that virus has. So if we say that something is H3N2, it's got version 3 of the H spike, version 2 of the N spike. That's what the naming means. This picture is super important. I'm going to probably spend the rest of the time on it. This is a picture of H spikes of influenza. So you could label this. This whole thing is an H spike of an influenza virus. And you can see that they're poking out of the envelope here. It's one protein that's repeated 3 times. So see how there's 3 of them? This would be trimer, a trimer protein, and it's sticking out from the envelope. And up here is where it would be attaching to human cells. So something up here would be attaching onto the human cells and letting this virus into the human cells. These spikes have a certain region in the purple that let them bind to the human receptor. So this region right here, if we go, it would be the part of the spike that binds to the human receptor. that allows attachment. So you could make this say allows attachment, these blue. This kind of more middle part of the spike is the one, or is the area where people have antibodies that could bind and destroy the virus. So we haven't talked about antibodies yet. We'll get to that later in the semester, but antibodies are some proteins that are made by the immune system to attack and destroy previously identified foreign shapes. If we've seen a shape in our body that we're not part of our body, we will develop immunity against it, and we will develop the ability to make these antibody attackers that would bind onto this and help destroy it. So this is the part of the spike where antibodies would bind. So if you want to be the best flu virus, infect the most people, make the more flu virus, what would be helpful in that is to make a spike protein that is different from what the antibodies have learned. So if you could change the part of you that antibodies bind to, and they think that they've never seen you before, they're not going to attack you. But if the part that lets you get into the host cell stays the same, you can still get into the host cell. You need like the perfect kind of mutation here and not here. Mm-hmm.
So are naturally formed antibodies stronger than non-natural?
Like the monoclonals?
Sorry?
Are you talking about like monoclonal ones that, what do you mean by not naturally?
So you get like a vaccine, then you get like not natural antibodies. But like,
what you're getting in a vaccine is you're getting a piece of the pathogen, so that then you can make antibodies.
okay.
Yeah.
But like if you say like you actually had COVID,
Mm-hmm.
the antibodies stronger than say?
They should be.
Okay.
If it acts like a normal virus, they should be. We don't have enough data yet to know for sure. Like just in my own personal life, I've always got my ears pricked up for people who had COVID pretty badly last year, if they've got it pretty badly right now. So far, I don't know anybody. That's not data, that's just like me keeping my ears pricked up. But the question that you're trying to ask when we get to vaccines, which is that next week? I don't know. Like it's during this semester. I'm gonna come back to that question about like what makes a vaccine better than another vaccine. But if you wanted to be, the best, most effective flu virus, you would need to change yourself here, but leave this part alone. So here's the key to how viruses act. Viruses can't decide to do this. They're incredibly, I want to say dumb. Like they can't even be considered dumb. There's no brain. They're not even alive. They're just particles. But what ends up happening, what ends up being the most advantageous I'm trying to write the word advantageous. The most advantageous thing for a virus is to have mutations that change you enough to evade immunity. but still infect. That's what ends up being advantageous. And the reason that viruses are so good at this has to do with their genome copying mechanisms. Viruses have these weird, some of them have weird genomes, some of them have RNA genomes, some of them have a single-stranded DNA genome. And copying of those kind of genomes tends to be sloppy or mutation prone. So some viruses have a high mutation rate because they have sloppy copying enzymes. Some viruses mutate faster than others. Influenza virus mutates really fast, just constantly. It's got really sloppy genome copying. Coronavirus actually is not intrinsically a fast mutator. It's just that there's so many coronaviruses in the world right now, so many of them, that as a group, there's a high chance that they're going to develop the perfect mutation. So having a mutation that lets you hide from the immune system is going to be advantageous, and you're going to be the best, and you're going to spread to everybody, right? That's going to let you be the winner. But they don't mutate on purpose. So here's a dumb example. I'm going to describe genome copying. Go back to a place where we've got a picture of synthesis right here. Okay, so you're a virus and you infect the cell and the host is making a lot of copies of your genome pretty sloppily. And so imagine they're like papers. Like you go to my house and there's all these papers everywhere. All over the counter. This is kind of true right now. All over the counter. There's some over by the chair. It's homework, it's bills, it's junk mail. And there's all these different papers, and most of them are completely pointless to you. Like if you go in my house, you find a random paper. It's an Arby's coupon that expired last year. Woo. But somewhere in the papers of my house is my son's driver's permit temporary card. I think it's in his room. I think I took it to his room and put it with his papers. And like, if you found that one, that could be really useful. Like you could steal his identity. You could, I don't know. you could do some damage with that if you found that one. But most of the papers in the house are complete junk. So most viral mutants are complete junk. Or what we called when we did the little exercise with mutations, most viral mutants are going to be worse, way worse. They're not better infectors, they're worse. One in a billion mutants might be better. Might give you better infection, might hide from the immune system, but still infect the cell. It's so rare, it's 1 in a billion, but man, how many coronaviruses do we have on the earth right now? It's like a trillion times a trillion times a trillion. It's a tiny chance that any given one of them will be better. But when it's better, it wins, it infects more people. And it becomes the dominant strain like Delta is right now. Who was asking about mu? Is that this class? Somebody in one of my classes was asking about the mu variant of coronavirus. So there was alpha variant and beta variant. The C one like no one ever talked about it. And then Delta, which is really a great virus. And then there's one called MU, and like it's not even that great. It's not as good as Delta. Delta is winning. Viruses don't make the perfect mutation on purpose. It just happens in a crazy chance, and then it's so advantageous that becomes the dominant mutation. That's what I'm going to end on today, is that concept. When we start back on Monday, you'll still need these slides. If you haven't already printed the Monday slides, don't do that yet, because that's from last year, and I got to update things. A lot of things are updated from last year about coronavirus, so I will bring you all notes for printouts for Monday slides because it's a current topic.